All right, so thank you, Barbara and Gary, for the interaction, also for introducing the meeting. It's just a great pleasure to be here today with such a massive amount of brains in the field, which is actually quite important and quite interesting for us, because we actually rarely we join forces to actually work together in 3 different disciplines, but at the same time so common. So I'm going to be talking today about fungicide resistance in Australia, but I wanted to focus a little bit on what's the monitoring status. So from traditional monitoring to what is actually what we are doing now to speed up the process of monitoring diseases in the field. So resistance is something that actually is natural. So it's just evolution, okay? So here I'm just presenting 2 graphs. The left-hand side top is just showing what has been the progression of fungicide use together linked with the production of the yield of wheat in the UK. So actually there is a massive correlation between increased yield and also increase in fungicide usage. But at the same time, there's a correlation between the interaction of fungicides and the development of resistance. So we don't create resistance, we select for resistance, which is quite an important concept. So till now, pretty much, we've got around 350 cases of different species that have been found to be resistant to different compounds. And this is field resistance, not in vitro resistance. So fungicides are really important, obviously. So they are comparable in importance to breeding. So actually, losing a fungicide is losing a really important management tool. So in this other slide, what I'm presenting is the number of cases of fungicide resistance basically identified in Australia and confirmed. So far, there are a few. Actually, some of them are more concerning than others. But obviously, this number will increase with the power that we've got now to screen more isolates and to screen bigger areas and to have access to more samples. Traditional monitoring is what basically everybody does. So we do traditional monitoring because this is what we have learned to do, and because sometimes we don't have any other option. So sometimes it's just a matter of how many samples can I test or how many samples can I access. So everything starts with a report, fungicide failure, somebody having issues to control the disease, stuff like that. So it takes a lot of time to collect a number of samples that is meaningful, all right? So then we need to step into a different stage, it's a discovery stage. So we need to find out what's the correlation between the phenotype you find in the field and the resistance, okay? So, and sometimes it's very difficult and unsuccessful. So not every time we find a genotype that correlates with the phenotype. So when we finish with those stages, we enter in the monitoring per se. So monitoring doesn't start till then. So then you can just go and target areas that are particularly interesting because they full of a history of a spray record history, and a history, track record of fungicide failure. So it's actually a really time-consuming process. Example of this traditional monitoring is, for example, so botrytis in grapes. So you have a de novo problem. So you've got an issue from the industry, and you have to give an answer. So you try to find responses to fungicide failure in the field. So you need to create baselines, so with different isolates, different fungicides, and you need to find out what's the distribution of isolates in the population. So once you get to that point, well, you start analyzing, as I said, what are the genotypes. So in this table, I'm presenting genotypes or resistance to different compounds found in botrytis in grapes. So here, with this color code, you can see how resistance increases with the red. So you've got, sorry, because of the color, this was supposed to be orange, and it's actually dark yellow. So you've got strains that are resistant to 1 or 2 compounds, strains resistant to 3 to 4 compounds, and strains that are resistant to 5 to 6 compounds at the same time. Okay? This is a really long process. I'm not going to talk too much about this because there is another talk on fungus resistance on grapes tomorrow. So I invite you to come and have a look. So something that basically speeds up the process is the high throughput monitoring. So we know a genotype, so we already know what is actually responsible for this problem, so we can respond much faster. So we deploy baiting trials. So baiting trials are just trials with a specific compound specifically designed to catch disease and to catch mutants. So once you've got those waiting trials, you can do targeted monitoring, you can do targeted collection. So you collect there, and then you develop a highly sensitive and high throughput methodology to test those genotypes. There are many. So we've been focusing on one that is digital PCR. So when using this technique, we have been monitoring what's the situation with barley powdery mildew. So rather than now analyzing single spore isolates, that it takes a really long time to prepare those isolates, we do bulk sample analysis. So we use baiting trials combined with this sophisticated, highly sensitive technique, and we identify the different genotypes. So if the red bar is showing the situation where Australia was in terms of genotypes 5 years ago. So this is basically the progression of the different mutations found in Europe. Okay? So 5 years later, we were here. So suddenly, the whole population in Western Australia jumped to a status not seen before, much resistant than anything that's seen before in the world. All right? Responsible for that was that mutation, S509T. Okay? So it took us 3 years to get to that point, so to understand what was going on. It took us 3 months with this new high-throughput sampling methodology to understand what was going on in other regions in Australia. So now we know that it's not only Western Australia, that it's pretty much 100% resistant to the, so carrying this genotype. Now it's also Victoria, Tasmania, and to some degree, New South Wales. There is another thing that we call hybrid monitoring. So we don't know the genotype that we are after, but we can still deploy baiting trials. So we can do this targeted monitoring. So we don't need to go like crazy to the field and collect samples from everywhere and then process everything. So we can do this targeted sampling just deploying baiting trials. This is, for example, what we've been doing with barley net tight net blotch. So with barley net tight net blotch, we have baiting trials also across Australia, and we've been trying to understand why is it that it's resistant to certain DMIs. So obviously you have to create still these baselines. You need to understand what's the population response to the different compounds. And then once you've got a phenotype and you've got a strong phenotype, you have to try to correlate it with DNA sequencing. Obviously it takes time, but you know the targets you are after. So we have correlated that with this particular mutation. First time described in literature, first time that we see something like this. Because it's not only this mutation, and I'm showing here all the results of, for example, gene duplication and stuff like that. It's much more complex than that. But you need to go through that DNA sequencing stage. How am I doing with the time?
You have 3 minutes
left. All right. I also would like to call hybrid monitoring the issue with canola blackleg. So we've been doing some analysis on samples coming from Angela van den Boer and Barbara Holland. Basically, these samples, we had to send them through the same sort of process. We submit them through the same sort of process. We calculate baselines. And we know now is pretty much what's the response to each of these isolates to different compounds. All right? So it's interesting for us and probably for the industry as well, because some of these compounds are not registered. But something that came out quite interestingly is a particular isolate being extremely resistant to apridium. The reason why it says more than 10 is because that was the highest concentration. In further testing, we know that it's more than 150. So it's actually pretty much insensitive. So obviously, blackleg is not, apridium is not registered for blackleg. It's a sclerotinia. So obviously, the fungus doesn't know that it's not being, target disease, but still it's treated with a fungicide. So you select for resistance, even though you are not dealing with the pathogen with a spray application. Again, we have to correlate that with the genotype, and this is what we found. So this is another mutation, new mutation, the target gene of apridium OS1 that is extremely similar to a mutation found in Botrytis cinerea. So it's not actually a surprise. In the end, if I want you to basically take home a message from me, it's just monitor, monitor, monitor. This is something that we've been running for a while. We ask people to provide samples. We ask people to get in touch if there are fungicide issues in the field. And we ask people to collaborate with us in trying to find responses to this. All all this work that is just a very brief summary of what is going on, has been happening in the fungicide resistance group at the CCDM at Carleton University. But we don't do this alone. Many people work on this together, different collaborators from Western Australia and especially in the Eastern States. Obviously, the money payers, really important. So they pay our bills, they pay our salaries, and our experiments. So I think I'm going to leave it here. So if you've got a question, I'll be more than happy to try to answer.
Thank you. Thank you. Fran.
I've got another presentation if you guys want. I can just...
Okay, I'll ask a question then. When you spoke about the barley powdery mildew, you talked about a range of mutations there, but you didn't tell us which particular gene it was in. Is that in the cytochrome P450?
That's the CYP51, yes, okay. Correct. Yeah.
And the target one, the iperidione, what is the target gene
That's
there?
OS1. That's OS1. Target gene is OS1. So it's
OS1,
yeah,
which is?
It's basically a MAP kinase.
It's a MAP kinase.
Pretty much,
okay.
Yeah.
And is that the only target? Well, you've said that it is for that particular fungicide.
There's another potential target that is OS2.
Yes.
It's actually quite correlated to this one, but it's slightly different. So you can find mutations in OS2 as well, but primarily in OS1. Yes.
Okay. Other questions, please? Yes.
Friends, I have a question for you. So in addition to the CYP51 mutation, there's any other weak resistance mechanisms in fungicide resistance?
Yes, there are plenty of them.
How can you monitor this free resistance?
So you can do, there are many things that you can do there. I mean, everything relies on collecting samples, obviously. But, I mean, if you are talking from the point of view of the lab, what can we do in vitro? So there are certain tests that you can run. Okay, so if you're looking after, for example, an ABC transporter overexpression, so obviously the test is going to be more difficult than, looking for a mutation. At the end of the day, if you correlate that overexpression with changes in the promoter, you can do exactly the same. You just detect mutations or SNPs or, indels. It's not a very difficult thing to do. And this is basically what I do. So I invite you to go to a couple of talks that are going to be run tomorrow during the fungicide workshop on these detection techniques, so especially digital PCR and LAMP. So there are many things that nowadays we can do, and that we should start deploying in the field as something normal. So I think that we rely too much in traditional methodologies.
Got a question across here? Yes, at the back.
If I've read that right, my powdery mildew resistance has come over from the west to Victoria, South Australia.
That will be difficult. I mean, we need to run this. That we have the same sort of question at the moment. So we need to run some microsatellites testing on samples to try to find out, where the epidemic is coming from. I mean, powdery mildew develops mutation very quickly, and under the right stress, it's going to develop the mutations. So this mutation is happening just because it's the most likely to happen without casting a massive fitness penalty to the species. So all the isolates in Australia carry already a mutation that we call the precursor mutation. So it's just per se, the wild type already carries it. There is no wild type here. So obviously that mutation is predisposing strains to further mutate. So that mutation.
Into the mic, sorry.
Oh, sorry. Yeah. Oh, sorry. Yeah.
No, it's mic. I didn't either.
Do I need to repeat the whole thing?
No.
All right.
I've got about 10 seconds.
10 seconds left. All right. So probably we should leave it for further discussion later on. But yeah, so the mutation, it could be happening. It could be coming from Western Australia. Most likely that they have developed in the United States already. Okay.
Okay. Thank you very much. Gary's just pointed out to me that this is being recorded. So even though we can perhaps all hear, the people who are listening elsewhere may be not. So therefore, people, please put these on. Another thing is there are other seats here. You don't have to stand up unless you want to. Okay. Our next speaker is Andrew Milgate. Andrews from the Department of Primary Industry, New South Wales.
Thanks, Barb. Firstly, thanks to the organisers for the opportunity to come and speak today to you about the work we've been doing on Zymo septoria tritici, or otherwise known commonly as Septoria tritici blotch. We've only been working on this issue of fungicide resistance in this pathogen for the last sort of 18 months to 2 years now. So we're at the beginning of the discovery phase, and so some of the results here are probably similar to what Fran was putting up, but in earlier stages of the evolution of our knowledge. Today I'm just going to run through where septoria is a problem for us today in Australia, a quick overview of the modes of action that we've got available in terms of the fungicides, what we're finding in relation to the isolates that we've been looking at and where they've come from, and some of the consequences of those in relation to comparing the results that we've seen with overseas results as well, and where do we go from here in terms of managing this situation. So Septoria tritici blotch is a hemibiotrophic pathogen, so it causes necrotic lesions after an initial infection period and then an extended period of causing no physical symptoms on the plant, which can extend up to 21 days. It then causes necrotic lesions to develop on wheat plants. It can cause up to 50% yield loss in susceptible varieties in conducive conditions, and you can see here on this side in this genotype here that we've got complete loss of the leaf area versus the other genotype with some level of resistance. So where we've been, where's my timer, Barb? Can I see that timer? Thanks. So I know where I'm at. In terms of where we've been seeing Septoria re-emerge as a pathogen of significance in Australia, it's really in the southeast of the country. The red dots here indicate where we have isolated current collections from. Across the southwest of Victoria in the high rainfall and the southeast of South Australia in those irrigated areas down there, as well as right throughout Tasmania. I've also put some blue dots on the map there to show where historically in Australia this pathogen has been around since the 1940s. There's as far back as the records that I've got access to, and that extends right up into central New South Wales. It's largely a cool climate disease. It prefers moist, wet winters, and that has extended right across the southern part of Australia into Western Australia as well. Interestingly, even though we've seen a re-emergence of the disease in the southeast, in Western Australia we haven't picked up any significant populations developing over there yet. In Australia, prior to this year, we've been reliant on two families of chemicals in the fungicides for control of this pathogen and other pathogens in broadacre cropping. This year, and obviously going forward from now, the SDHIs are emerging onto the market as another tool in the fungicide makeup that we've got to use against these pathogens. So all of the fungicides that are applied in a foliar context in Australia have a triazole backbone, and some have the addition of a strobilurin, and that really is an important thing to remember in terms of how we utilise and rotate these chemistries in broadacre cropping for control of pathogens. Fran's already given a very good introduction in terms of the evolution of pathogen resistance to fungicides, but I thought I'd just drop in that we're really looking at two different modes of evolution of resistance to fungicides in different genes. The ones that we're primarily interested in with Septoria have been in the CYP51 gene, which is a single mutation event that occurs and it causes complete loss of sensitivity or becomes resistant with that single mutation, versus the CYP51 gene, which associates with the triazole group of fungicides, and that overseas has been shown to be being caused by an accumulation of small step changes in the mutations in that gene over time. So when we've sequenced the CYP51 gene of the isolates that we've been looking at in Australia, initially we started with 100 isolates dating back to the 1970s. We found the good news is that we haven't found any changes in the cytochrome B gene or that significant mutation there that causes breakdown of the strobilurins. But what we did discover was that we'd found 6 mutations and changes from the wild type in the CYP51 gene. Now, 2 of those have been implicated in causing some loss of sensitivity. They are consistent with the mutations that were discovered early in the introduction of triazoles in the UK and Europe, and so we were particularly interested in these. And then what we also saw by looking back at this historical set of isolates that we have was that it was the 2011 to 2013 collections where these mutations that were associated with fungicide resistance actually turned up. So just graphically showing that is that in terms of the number of isolates that we've looked at across the years in groups there, 3 distinct periods where we had all sensitive alleles in the 1970s and 80s and the early 2000s, and then a dramatic shift in 2011 and 12 where we saw the emergence of the 137F mutation and a small number of the 461S mutations, and then those haplotypes increasing in frequency in the samples that we looked at from 2013. And we've also picked up a number of isolates with a unique combination that hasn't been reported elsewhere of the 137F in combination with the 461S. So that's our sequencing data that's showing us those. We've then carried those isolates through to the lab bioassays that are outlaid in the FRAC protocols, and we've characterised their level of resistance to a number of the triazoles. So in this case, I'm just showing you the propiconazole, prothioconazole, and tebuconazole, and their resistance factors in the group of isolates that we've looked at so far. And you can see that we've discovered a range of reactions there, extending out in the propiconazole to above 20 times that of the wild type that we've been comparing our isolates to, and some variation in the tebuconazole and the prothioconazole as well. So how does this compare our results to what's been published overseas out of the UK and Europe? Well, in some ways it lines up well, and in other ways it doesn't line up at all. This table shows the significant mutations that have been discovered overseas and the resistance factors to a range of chemicals there, and I've highlighted the 2 important ones that we're working with in Australia. And you look at that data there and you think, well, the 137F is not a particularly nasty mutation, and it's not causing too many problems across there except for tridomenol, whereas the 461S looks like a more significant one. And the important thing is that we haven't picked up important mutations like the 136A in combination with the other mutations, nor have we picked up the 524T. And those mutations in the UK have elevated the level of resistance significantly, but we haven't, fortunately, we haven't found them so far. So when we look at those results, and then I'm a little, and look at the results that I knew we were getting out of our lab, and we should see differences falling out in terms of the sensitivity of those isolates to particular triazole fungicides. However, when we actually compare the reactions against each other, either propiconazole against tebuconazole or prothioconazole against tebuconazole, what we actually see is a pretty linear relationship. And what we also see in our results is that the 137F, which is supposed to elevate the level of resistance, in some cases, in some isolates, is much more sensitive than the wild types that we've compared, which is up here. So if we were basing our resistance factors calculations off our most sensitive isolate, we'd have much higher values. So there's some inconsistencies between the work that we've done and what's been seen overseas. So we've got another tranche of isolates that we'll be finishing phenotyping soon to add to this data set, as well as an additional set of genotypes to put together to see whether that changes that relationship. But at the moment, I would say that we're not seeing necessarily a good correlation between haplotype and elevated levels of resistance, but we are seeing significant changes in the phenotypes in the lab. So moving on to, just to finish up, in terms of the fungicide resistance management message that we've been promoting, particularly in the southeast of South Australia and southwest, across the southeast of Australia recently, is to try and get growers to think about reducing the overall population size and using the tools that they've got. And so in terms of fungicide resistance management, we've been trying to get people to think about that there's a regional level and a paddock level to address, and that there are certain levers that can be pulled to address that, and that if we can do that, that will help manage that situation overall. And that I'm just trying to keep to time here, so I'm cutting this a little bit shorter, but that those integrated disease management messages and fungicide resistant management messages are largely the same. It's just we want to try and encourage growers to think about fungicides not as the go-to thing, but to also understand that they can pull those other levers in terms of stubble management, timing of sowing and variety choice to complement their fungicide usage into the future, and by that way delay selection of more resistant isolates. Because at the moment, the growers are using what they believe to be the most effective fungicides. That's been used broadly consistently across the region. They're using it multiple times within the crop. And the population, the septoria population, has responded to that and is going to continue to respond to that if they keep using the fungicides in this way. And so we really want to push the integrated disease management message. I've put a question mark beside rotating fungicides, just because it seems to me that our results are a little unclear about the value of rotating fungicides at the moment, and that's something we're going to have to work on in terms of a value for farmers going forward. And monitoring, as Fran has also mentioned, is obviously going to be important moving forward in this space. And I'll just leave you with this picture that I took about a fortnight ago in Victoria of a crop where the farmer told me he'd applied two full rates of Prosaro, so that's prothioconazole and tebuconazole together on this crop, and I could still find lesions like this of septoria in the canopy. Now, it's possible that these are just escapes, but it's also possible that these are the types of isolates that are going to be the next evolution in fungicide resistance in this pathogen. Okay.
Thank you. Questions for Andrew? Yes.
Andrew, Sue Cross from Mayo. Resistance factor for triazoles, I believe in the case of pathogens like soybean rust, a resistance factor of up to 10 is considered inconsequential or within the accepted range. Is that the same for
Yeah, well, that's an issue that I've been really interested in terms of trying to relate, how do we relate those lab results to field practice and what's actually functional in the field. And we've been working on developing an assay to test that in the glasshouse. And it looks to me as though, and I'll be showing these results tomorrow in the talk tomorrow, that even small changes in those EC50 values are translating to significant survival advantages in the field for the pathogen, and that may be as low as EC50 values increases of, or resistance factor increases of 1 or 2 above the wild type do appear to provide additional survivability in the presence of fungicides in planter.
So on that point, is there any, do you have any evidence of any fitness penalty for any of the mutations?
No, not at this stage we don't. It would be interesting to know. We've got a number of collections that I think that it will be interesting to examine that, where we've got where no fungicide has been applied for 2 years in that southwestern Victoria area. So to compare with other paddocks populations that we've collected where there's been continuous application of fungicide. So it will be interesting to see what they reveal.
Andrew, I think your comment about disease escape is really important. We get a lot of reports from farmers and agronomists saying, you know, fungicide hasn't worked, but I think in a lot of cases disease escape can explain the situation. And I guess understanding when disease escape is important versus fungicide resistance and tolerance becoming a big issue for industry, I guess.
Repeat the question.
Not really sure how we address it, but it's something that we need to keep in mind in terms of screening in the lab versus what's going on in the field.
Yeah. I think that, okay, so I've been asked to repeat the question. I think I'll run the mic up to you. So I guess the question is about the importance of disease escapes for industry versus actual real, evolution of fungicide resistance and the frequency of that happening. Look, I think that with the vagaries of timing of application being so important in the disease cycle and the expression of disease and the effectiveness that you can expect out of those fungicides in the field is massive and completely, well, not completely, but largely misunderstood by the grower community. So yeah, I'm more putting that up as that these are the types of things that, as Fran was alluding to in these trap plots, these are the types of things you are switched on to being interested in from a monitoring point of view. But yes, that understanding the limitations of fungicide application in the field and how to best manage that to minimise escapes is a big issue. Yeah.
Thank you very much, Andrew. Our next speaker is Paul Ruminer from the University of Melbourne.
Thanks, Barbara. Good morning, everyone. Look, today I'm going to talk about some work that we've been undertaking in our lab and working with collaborators over a number of years, working with 2 species, the green peach aphid and red-legged earth mites, which are both quite important broadacre pests. And I guess through the talk, just talk about some of the contrasting stories between these 2 broadacre pests in terms of both, I guess, particularly the spread of resistance, but also to a lesser extent the evolution of resistance in these 2 species. For the non-entomologists in the room, just very briefly, the red-legged earth mite, first and foremost, is a species that we find widely distributed across Australia. In the field, it reaches very high densities, in excess of 20,000 mites per square metre, and economically, estimates have put this particular species at causing in excess of $500 million per annum to Australian agricultural industries. The second species, the green peach aphid, well, it's a species that attacks a variety of different crops, not just in broadacre, but across a number of different industries. And like many aphids, it transmits a number of important plant viruses, including one that I guess many of you will have heard about more recently, which is beet western yellows virus, given that in 2014 there was quite a severe outbreak of this virus in southeastern Australia. And some estimates of that particular outbreak put the yield losses of in excess of 150,000 tonnes. So I guess you can see both of these species economically very important pests for the agricultural and the broadacre industry. And of course, from a resistance point of view, both of these species have insecticide resistance, and those resistances, some of the issues associated with that appear to be escalating. In the interest of time, I don't have the time, I guess, to run through this table step by step, but I guess what I've just demonstrated here is some of the key biological traits for the green peach aphid and the red-legged earth mite. And I guess the point I'm really trying to make with this slide is that these 2 species biologically are quite distinct in terms of their mode of reproduction, the number of generations that they have during a calendar year. Their host range is also quite different. Their distribution, both within Australia and outside of Australia, varies considerably. And also, importantly, from a resistance spread point of view, their dispersal mechanisms are also vastly different. So I guess, we have two species that biologically are quite different, but what does that mean from a resistance point of view? And I guess, really, most importantly, what does it mean from a grower's perspective in terms of, the situations and the issues that are going to arise and are arising currently? So firstly, if we just look at insecticide resistance in the red-legged earth mite, resistance was first confirmed fairly recently in 2006 at a single location in WA. Resistance was found to one chemical class, the pyrethroid group, but to no other chemicals at that point in time. We saw very high levels of resistance in the field, in excess of 200,000-fold level of resistance, and since then, there's been a novel mutation in the parasodium channel that's been identified to be responsible for that very high level of resistance in this particular species. So since that time, we, working together with CSIRO and Department of Ag in WA in particular, but also other state agencies around the country, have been undertaking some pretty extensive surveillance activities of resistance in the field. And this map here essentially just shows what we currently think is a pretty accurate depiction of the distribution of resistance in WA now, as we, I guess, currently stand. So as you can see, we now have resistance spread right across the WA greenbelt. Some of these populations, are well over 1,000 kilometres apart. The good news, I guess, for us on the East Coast is, despite pretty extensive sampling across a very large geographical area over a number of years, we've failed to find any evidence of resistance outside of WA. We have a new molecular diagnostic that's come online in the last couple of years, which of course has helped us be able to achieve, I guess, these very extensive surveillance activities, I guess similar to what we've already heard from Fran. But also importantly, it's allowed us to develop a diagnostic service that's been made available through GRDC directly to growers. And that's something that, particularly in WA, growers and agronomists are actually taking up. So we've got a really valuable tool there that's helping us both from a research point of view, but also directly in terms of advice and information in real time for growers. I guess one of the more recent developments that's occurred within this story is that last year, unfortunately, we had the first case of organophosphate resistance popping up in this species, and this was found at a couple of sites in Western Australia. And I guess this discovery in particular is really, I guess, a little bit concerning, given that for those that don't know, currently there are only two chemical classes registered to control red-legged earth mites in the field as foliar applications, and they are organophosphates and pyrethroids. So we are now faced with a situation where both of those chemicals are breaking down, and of course going forward, there's an imperative that we understand these resistances and that we can really develop some solutions for growers going ahead, so that we've got tools available into the future whereby we can get control of this pest. One of the other things that we've been doing as part of our project has been trying to understand the localised spread of resistance. And this is a slide that I certainly don't have the time to fully explain. But what we've been doing is going to multiple sites in WA where resistance is known and trying to monitor quite at a very fine spatial scale repeatedly through time to get a handle on how quickly or how slowly resistance alleles are actually spreading out from these populations. And so one of the things that's been undertaken is some transects have been ran out. This is at a site near Cranbrook. So we've ran some transects out, reaching out to about 1 kilometre away from resistance paddocks into adjoining farms. And mites have been collected at every 100 metres away from that point. And those mites have then been, I guess, assessed for their presence of resistance alleles. And in this particular transect, you can actually see in this graph here, the corresponding graph, that there's actually no movement of resistance alleles outside of that particular paddock. A transect running in the easterly direction, just another example, if you can see this map, this graph here. In this case, we do see around 20% resistance alleles moving out, but that pretty quickly drops off back to zero, so to full susceptibility. So I guess really it does just illustrate that, at least at a local level, we have fairly slow movement of resistance happening in this species. Of course, we are also seeing evidence of jumps, though, and perhaps through human-mediated dispersal. So for green peach aphids, I guess historically we've had both organophosphate and pyrethroid resistance in Australia for quite some time now. but in 2010 we had the first documented case of something called the MACE resistance, which confers resistance to a third chemical group, that's carbamates. So for those that know a little bit about insecticides, so that's products like pyrimicarb. And so I guess off the back of that, that discovery, we led, again, with a number of different project collaborators, I guess an extensive effort to collect and then look at the resistance frequencies or the presence of field resistance in green peach aphid populations across Australia. Again, I'm going to whip through this relatively quickly, but we have 3 maps down the bottom here: organophosphates, carbamates and pyrethroids, so these 3 chemical classes. And if you just sort of scan your eye across these maps pretty quickly, you'll see that we've got some red circles and green circles. The red circles indicate where we found these populations to be resistant to these chemicals, and the green is where we've got susceptibility. And as you can see, this work that was carried out in 2012 and 2013 showed that certainly we had resistance that was widespread across Australia, but I guess importantly, and particularly for the organophosphate group, we saw a lot of populations that were still susceptible to these chemicals. So this was the story at the end of, I guess, 2013. And as I mentioned earlier, in 2014 there was quite a large outbreak of beet western yellows virus caused by or vectored by the green peach aphid. So as a consequence, we actually subsequently received quite a large number of samples from growers and agronomists and researchers, principally from southeastern Australia, but we also took it upon ourselves to get samples from elsewhere in Australia. So since this time, we've actually screened in excess of another 70 populations nationally. And these are the results for the work that we've recently completed, looking at populations collected in the spring of 2014 and I guess earlier in the year, this year. And immediately what you'll see, this is a map for pyrethroids, is that all of a sudden, the large number of our populations have 100% resistance, and there's not a single population that's susceptible. This is the map for carbamates, so the second chemical class, quite a similar story. Most populations are 100% resistant and none that showed full susceptibility. And organophosphates, the third class. So I guess really what I'm demonstrating here and what was a little bit surprising to us was just how quickly we could see a pretty dramatic change in field resistance in this particular species. So within a matter of sort of 18 months to 2 years, we've gone from a situation where we still had products that were going to be working in the field to a situation where we now have widespread resistance across green peach aphid populations. So it really shows, at least in this species, how quickly I guess we can get this movement of resistance dominating and moving across whole landscapes. Some of the other work that we've been involved in with green peach aphids is looking at, I guess, getting at that question of movement and importance of resistance being transferred from one area to another. And so we've got at some of these questions using some population genetic processes. So what we've done, again, looking nationally, is look at a whole variety of different green peach aphid populations. And what we've found, given that this species reproduces asexually, is that we've got these unique clonal types that are found within Australia. To date, we've identified about 40 unique clonal types, and we've screened thousands of individuals. So we've got a pretty comprehensive data set here. And these unique clonal types have different distributions and different resistance profiles. Some of them are susceptible to some chemicals and some of them are not, or at least historically they were susceptible. I guess the point I really wanted to make, though, was what we've found, which is, I guess, quite interesting and potentially explains a lot of what we're seeing in terms of that movement of resistance across Australia, is that we've got 3 superclones that we've identified. So 3 clones that make up in excess of 80% of all green peach aphids within Australia. And we're finding these clones to have very wide distributions across Australia. And each of these clones is resistant to the pyrethroids, is resistant to the organophosphates, and is resistant to the third chemical class, being carbamates. So I guess that's a recent field development for this particular species. So of course, as Andrew and both Fran have alluded to, monitoring and understanding what's happening in the field is critically important. As scientists, it's very interesting to us, but critically, the reason we're doing this is so that we can inform resistance management and we can get practice change happening. And so the last part of my talk, I really just wanted to talk about this, I guess, this concept of resistance management in terms of insects and particularly these 2 species. So in 2013, GRDC established a group called the National Insecticide Resistance Management Group, or NIRM, and NIRM's responsible for developing resistance management strategies for a number of key pests. And you know, the group's been operating for a couple of years, and I guess, and again Andrew alluded to this, one of the initial questions or challenges that we face when dealing with these discrete pests in terms of resistance management is what is the appropriate spatial scale in which we should operate. So the first resistance management strategy produced by this group was for green peach aphids, and this was developed last year. And of course I don't have time to run through the details, but what I will just point out is that in this instance, based on what we know about the biology of the pest and obviously what we know about the resistance that we're seeing across Australia, the spatial scale that we've operated on in terms of recommendations for how we might rotate chemicals, for example, has really been looking at a very large level. So we've actually got recommendations looking at state practices. Of course, we've seen quite a different story emerging for red-legged earth mites, and NIRM tackled a resistance management strategy for this species only this year, and it's a draft at this stage, but it will be finalised in the next little while and published next year and hopefully endorsed by CropLife Australia. And again, I'm not going to go into the details, but just to point out that in this instance, the decision by NIRM is really to put forward recommendations at a paddock level. So quite a contrasting view around how we might manage these two species going forwards. So in the interest of time, I'll cut it short there. But certainly just do want to acknowledge the work that I've presented is from a large number of different organisations, both within our group and outside. And of course, just acknowledge the funders of both GRDC and Horticulture Innovation Australia.
Questions for Paul? Yes.
Paul, the three clones that you said that are resistant to the three different insecticides, do you think they're independently derived resistance, or do you think there's been some kind of sexual combination in the clone's history?
Yeah, look, to be honest, right at this stage, we're not quite sure. We're just getting at the bottom of those sorts of questions. I mean, obviously, we can get at that looking at the microsatellite data that we've developed, but you know, we just haven't had a look at that just yet. But of course, you know, we will probably hear from Tom Walsh, I suspect, who's going to also talk about green peach aphids and some of the genetic work that we're doing collectively with CSIRO. And of course, there's also the possibility of incursions of new biotypes coming into Australia. And that's also something that, you know, is critically important to this whole story of resistance in this species. But I'll let Tom talk about that, I'm sure.
Any more questions? Yes.
Yeah, so, okay, the question was the organophosphate-resistant populations in red-legged earth mites, whether they are also resistant to pyrethroids. The short answer is yes.
Okay, thank you very much, Paul. I've got 2 timers there. It's very technical, isn't it? Okay, our next speaker is Grant Heron, and he's from DPI New South Wales.
Can I start it? That's a text page, so we've got to go to a presentation view. So click on that. So that's it. That's a PowerPoint. Now, how's that look? Look okay? Right. I'm softly spoken, so I do need a microphone. Thank you, ladies and gentlemen. So today I'm going to talk about resistance in secondary sucking pests of Australian cotton. I suppose it's, you know, why would I be talking about resistance in the secondary pests of cotton, and why not the major pests? Well, it's an interesting conundrum that because we're not spraying as much in cotton as we once were, the pests that were once secondary pests are now major pests. The whole pest system is changing, and it's because we're not spraying as much. So, because of this, the bugs, particularly the sucking pests, have become more dominant. And I want to talk about today about two-spotted mite. Two-spotted mites there on your left. That's a major pest of agriculture generally. It causes huge amount of damage. When it gets into cotton, it gets uncontrolled. It'll defoliate it. It makes a mess. On the right-hand side, you've got cotton aphid. It's also called melon aphid. It gets into all sorts of agricultural systems, but as with lots of aphids, it transmits viruses. And if you put virus transmission in conjunction with resistance, it just makes a mess of systems. In cotton, it transmits a virus called cotton bunchy top virus, and because of that, growers tend to want to spray it. It makes them nervous. So how do we go about doing this? So I do resistance work. I'm a resistance specialist. And like as a lot of the people that have been talking before me, we do molecular techniques like PCR sequencing, where we're looking for single nucleotide changes. But really, it's underpinned by the bioassay. And on that left-hand side there, you can see one of my research entomologists there, Kate. She's using a potter spray tower, and she's putting insecticide down that spray tower straight onto insects, which are on little leaf discs. And it's a phenotypic response. It's very powerful because you don't need to know the mechanism. It actually underpins everything that we do. So if we work on to two-spotted mite, everything that we're doing on two-spotted mite at the moment I'm doing via bioassay. And you can see there I'm bioassaying a number of chemicals: bifenthrin, abamectin, propagite and diphenthioron. And some of those are showing resistance. So we've got resistance to bifenthrin, abamectin and propagite. Bifenthrin is very high level resistance, and it seems to be that it's related to the way there is bifenthrin being used in cotton. Abamectin resistance, again, very high level resistance, and it's probably because abamectin's been put in with other sprays to control mirids. So it's concurrent selection, just kind of accidental use. And propagite there is the mainstay miticide for controlling mites in cotton, and there's incipient resistance there all the time. And the interesting thing is not that two-spotted mite is resistant, because two-spotted mite is resistant all over the world, but the mite complex is changing. Once two-spotted mite was the dominant mite in cotton, but now when you start looking at two-spotted mite, it's not there as much as what it was before. Even though it's resistant, it's not getting the selective advantage you'd expect it to have, and it seems to be being displaced by bean spider mite and strawberry mite. And I've got actually an entomologist working on this to try and sort it out. What I'd really want to do with two-spotted mite is move on to DNA-based methods. And to do that, I've used strains that I've collected. So that some people collect stamps, I collect resistant strains. That's what insecticide resistance people do. And I challenged those resistant strains using what we could find trawling through the literature about known resistance markers in two-spotted mite. And it's interesting that if you look down that list, you've got pyrethroids, carbamates, organophosphates, abamectin. They're the sorts of things that you'd see in cotton. I put in etoxazole and acequinazole just because I could, and we got a hit on etoxazole. So that was something that came in from horticulture, which is nice. You look at that, the mutation in red. So there might be a scientific note in there, so that was nice. And interestingly, although we've got heaps of abamectin resistance, and like most of the strains are resistant, we're not getting any. The mutations which are there that we know about are not involved in the mutations in my populations. Organophosphate, the bioassay is matching the molecular diagnostic. Everything's looking good. If you move up to the pyrethroids, the one on your right-hand side there, the bioassay matches the molecular diagnostic. Bioassay is resistant. The molecular is telling me it's resistant. Both those things go together very nicely, as did all the other ones. But unfortunately, when you move to this next one here, it didn't work so well. And this result relates to our reference two-spotted mite strain CD, which is pressured with bifenthrin and bioassay is highly resistant. So it is highly resistant. And of the 3-point mutations screened against CD, we only identified the F1694I mutation in the sodium channel gene, which is on the left-hand side. And this mutation was not found in our reference susceptible strain, which is the centre chromatogram. And given that this mutation is associated with pyrethroid resistance in the two-spotted mite strain, we thought we'd identified the target site mutation conferring resistance in that strain. However, after bioassay of the strain at the bifenthrin discriminating dose, individuals that were dead and therefore assumed to be susceptible were sequenced, and surprisingly the I1694 mutation was found, the right chromatogram. So what do these findings suggest? Does it mean the mutation is present in the susceptible individuals and therefore not a reliable indicator of resistance, or the individuals that died were just a control mortality anomaly? So what am I going to do? So there may not be a link between the phenotype and the genotype. Maybe it's detoxification. So I'm going to do some crossing. So if you go back to remember your Mendelian genetics, if you get a susceptible and a resistant and you cross it, you produce an F1, you back cross it to the susceptible, you're going to get a 50-50 split. So we're going to do that. We're going to bioassay it. We're going to run the molecular against it, and we hope we're going to get a 50-50 split by both methods. All right. What does it mean? Well, it tells me that you need to be very, very careful when you're introducing a new molecular diagnostic, and you need to back it up with your bioassay properly. And if it turns out to be a detoxification, well, they're oh so complicated, and much more needs to be done to verify it if it's a detoxification, and that's where more research needs to be done in that particular area. All right, mirids. Mirids are actually where where most of the insecticide that's going onto cotton is going at the moment. But unfortunately, there is no reliable bioassay to verify resistance in mirids at the moment. So what we did, we went trawling the literature, and we found something that was similar in another species, and we've used that, and we've included it, and that's a chromatogram there for the susceptible. And it's showing that our mirid strains that we're bringing in from the field are susceptible. But I wonder, does not finding resistance mean anything? We've separated the molecular diagnostic from the bioassay. Maybe it's a completely different resistance mechanism and we're just wasting our time. We don't know. Another thing, just you need to be careful. Onto cotton aphid. So cotton aphid, we're doing it two different ways. We're doing bioassay, and I've got my sample size there. We're testing 160 insects, and you're testing a range of compounds. So we've got their clothonidin, thimethoxam, diphenthyrine, and newly introduced sulfoxaphor. And interestingly, I'm not testing spirotetramat, which is new, or flunicamide, which is new, and I'll get to that at the end of the talk. We're using PCR. We started off using PCR, but I was unhappy with the sample size, and my molecular people introduced qPCR, so we're doing 200 aphids, which is a better sample size, but still it's not very good at delineating low-level resistance, so more work needs to be done on our methodology there, and I'm looking at two genes, so the ACE gene and a sodium channel gene. And in the ACE gene, we're looking for a pyrimidine-specific resistance, but actually confers cross-resistance to dimethyl-8 and O-methyl-8, and general OP resistance, such as chlorpyrifos or profenifos or something, that's at A3O2S, and pyrethroid resistance. So if we go on to pyrimidine resistance, if you look at that, the diagram on the right-hand side, you can see pyrimidine resistance turned up very quickly, which was what Paul was saying, and within 3 seasons, 96% of the strains were highly resistant. They were out of control. By 04-05, we started to get back control. So how was this done? It was done by sound resistance management, and by that I mean chemical alternation, windowing insecticides which were likely to cause cross-resistance away from the chemicals that you're trying to protect, limiting chemical use. But what really made the difference was the integrated pest management that went with it. And things like, reduce overwintering hosts, because the aphids would survive on overwintering hosts. So if you kill them, you put the resistant allele at a significant disadvantage. Other things, just like maintain beneficial insects. So you use selective chemistry. Sample effectively for aphids. Like every aphid that goes in your crop doesn't have to be sprayed. Make sure you've got the right species. And observe your thresholds. Spray when you need to. Like your chemicals that you spray should be last resort. You should be relying on your other IPM strategies before you spray. And then neonicotinoid resistance. You solve one, another one comes along. And again, it was underpinned by a sound resistance management strategy, but a little bit more. I've got 55 seconds. Is that correct?
Keep going.
Underpinned by a sound resistance management strategy, but more complicated because the neonicotinoids do more than just control aphids. And the difference is that here that 95% of the seed used in cotton is sprayed with a neonicotinoid. So why we're getting these fluctuations here is how it's used against pests other than neonicotinoids. So sulfoxaflor, here's my baseline data for sulfoxaflor. This is baseline data, and you look on the left-hand side, you've got the squares in the middle are my susceptible strain. What to the left of the squares is my least tolerant strain. To the right is more tolerant. I've got a good discriminating dose. However, despite that, we're still getting I've got sulfoxaflor survivors in the first year of the chemical use, which means that sulfoxaflor is being overused, and because of that, I reckon we're going to get resistance problems. The last bit was that here, new chemicals don't work the way chemicals used to. We used to spray organophosphates, carbamates. They work really quite quickly. Here's a chemical of that top graph. You've got flonicamid. In 24 hours, it's doing nothing. In 48 hours, it's doing a little bit. And if you look at the triangles, in 72 hours, I can't kill it. The methodology is not working. If you look at the bottom slide, again, we're at 72 hours. The methodology is not working. Even in increasing the temperature, the methodology is not working. So I can't test those chemicals using established techniques. And if monitoring underpins everything that we do, and if you can't effectively manage what you can't measure, we can't manage resistance unless we can sort this out. And that's where I finish. Thank you.
Questions for Grant? Yes, Steve.
Thank you, Grant. The advantage of this meeting is having people across insecticide, herbicide and fungicide resistance. And you very nicely pointed out that molecular diagnostics can only identify known resistance mutations. And we have really learnt that in herbicide resistance, where there are many mutations. And perhaps it's for the question session a bit later, but I just wonder, with the talks that preceded yours, whether relying only, I think is what you were saying, relying only on molecular diagnostics tests can really miss big existing resistance mechanisms and claiming, not claiming, but finding that populations are susceptible when they're actually...
Well, that was the point I was trying to make.
You certainly made it well.
Mm-hmm.
Yes, no, I agree. I also think that's something we should explore in the discussion. That's a point I really picked up on as well. Are there any more questions for Grant? Yes, at the back.
Yeah. Do you think that the reverse in, or the drop in resistant types of cotton aphid could also be emulated in something like green peach aphid with population?
If you have the appropriate strategies in place, I see no reason why not.
Any more questions? Okay, thank you very much, Grant. And our last speaker is Krishna. Sorry, she's standing in... Mahima Christian. She's standing in for Chris Preston, so it's a combined presentation. Thank you very much.
Good morning, everyone. Chris was supposed to give this talk, but had to pull out last minute because he's got a meeting. He will be here in the afternoon, so if you have any questions, please see him afterwards. I'm the new postdoc in this group, and my background is not weed science. I've been in this role for a few months, so I'll try and get through the questions if you have any, but I might handball them to Chris. So this is more of a status update about the herbicide resistance that we found in the field in Australia. A lot of this work was done with the random weed surveys that was funded by GRDC, and Peter Butsalis also helps out with a lot of these surveys. So this is not supposed to be comprehensive. It's just we'll just be highlighting some of the major sort of developments in the field in terms of herbicide resistance. So glyphosate is a very important herbicide in Australian agriculture. It's a broad spectrum herbicide, and because of its extensive use not only in the agricultural situation but also in other sort of situations, we've seen, not surprisingly, the development of resistance to glyphosate. The first instance of resistance to glyphosate was in ryegrass, and that was in 1996, and since then we've had 9 other species that have developed resistance to glyphosate. And in the past couple of years, we've also seen a couple of species that have developed resistance, one of them being the sweet summer grass, which we've identified in Queensland, and we've got a few populations of sow thistle from northern New South Wales that have also developed resistance to glyphosate, and these species have been found in summer fallows, which glyphosate is often used to control germination of weeds, often several times a year. And sow thistle especially is a problematic weed because the seed dispersal is through means of pappus. So pappus is just the fluffy stuff that you see on dandelion, which helps the seed spread readily between and within paddocks. So this is a dose-response curve of a glyphosate treatment at various rates to our treating our sow thistle species. So we've got here our susceptible plants, and then we've got 2 of our resistant populations. So sow thistle is not a very hardy weed, so it is quite susceptible to glyphosate, but the resistant plants need about over a kilogram of glyphosate active equivalent. So they are developing resistance, and we're just in the process of trying to understand the means of resistance of sow thistle. The other alternative to glyphosate is paraquat. Paraquat is also very important to control grass weeds in crops, and in this scenario we've seen 3 species that we've identified this year as having resistance to paraquat. And paraquat is not a very easy herbicide to develop resistance to. So these plants are machines. Some of them actually show no damage with resistance to paraquat. So one of them is crowsfoot grass, which shows the typical burnback symptoms that you get with paraquat treatment, but the undamaged tissue does grow back, so these plants have some resistance. We're, again, repeating these experiments and seeing how they are in subsequent generations. Pennsylvanian cudweed has probably been the most interesting one because there is complete die-off with susceptible plants, but with the resistance, there is very little damage that you can see. There are certain necrotic spots, but otherwise, if you didn't know that the plants were sprayed with paraquat, they actually look like quite fine, quite healthy. The other species is blackberry nightshade, which is not as resistant as the cudweed, but it's also a little bit more resistant than indica. So you do get a bit of tissue damage, but the plants generally tend to survive and perform okay after the treatment. So all these 3 species have come from the same region, from the same farmer, from Queensland, and this has been found in sugarcane crop. So paraquat is often used in sugarcane during pre-planting and also ratoon, and also used when there is no sugarcane being grown in the area. So the next herbicide is 2,4-D. 2,4-D is an auxinic herbicide, so it's a hormone, and it's the main herbicide that's used to treat broadleaf weeds, and especially it's become important in the recent days because a lot of the broadleaf species have developed resistance to Group B herbicides, which is the main herbicide to treat broadleaves. And so there is a greater reliance on 2,4-D to treat these broadleaves. So we've got some populations from southeastern South Australia and southern Victoria of south thistle. They're not hugely resistant at the moment, but this could change with prolonged use of 2,4-D over time. So you've got the susceptible plants here, and the resistant plants require about twice as much 2,4-D for their control. And from a practical point of view, the south thistle are not only resistant to 2,4-D, they're also resistant to other Group I herbicides. So here you've got plants that have been treated with 2,4-D, the resistant, susceptible, clopyralid. Again, here you've got the resistant, susceptible, and fluroxypyr. The resistant plants seem to be more tolerant to clopyralid and fluroxypyr than they are to 2,4-D. And this poses a big problem because we have very little to work with in terms of broadleaf control, considering that Group B herbicides have all been developed resistance to. We've got another member of the thistle family, capeweed, which has also developed resistance to 2,4-D, and it's also fairly low-level resistance. So they do get affected when you apply the 2,4-D. You do see the symptoms that you normally would with susceptible plants, but these plants tend to overcome that phase of stress and develop seeds and disperse them. And of course, capeweed being a part of the thistle family also disperses seed in the same way that sow thistle does. So once you get to seed dispersal, it's pretty hard to stop and control. So ryegrass has pretty much developed resistance to all post-emergent herbicides. So we're having to rely more on pre-emergent herbicides to control ryegrass in crop situations. We've got some samples from York Peninsula and in the mid-north of South Australia in the grain production fields, and they've developed a resistance to triolate, which is a pre-emergent herbicide. It's a Group J herbicide. And these plants also show some cross-resistance to prosulfocarb, which is the Boxer component of Boxer Gold. And there is some control that we get with Boxer Gold of these resistant ryegrass populations, but conditions have to be perfect. So these work okay in the lab under glasshouse conditions, but Boxer Gold might not be as effective in field conditions when the environment is a lot more variable. So this is a test that we've done with the ryegrass that we found to be resistant to triolate. So you've got the susceptible lines here, the resistance here, and you can see that the resistance increases from Boxer Gold all the way to triolate, and there is a little bit of damage. These plants do, I think, eventually die off, but prosulfocarb doesn't seem to control these ryegrass. We do get control of this ryegrass with pyroxasulfone, which is the active ingredient in Sakura, which is a Group K herbicide. And these are some other populations of ryegrass that have been found to be resistant to Avodex Extra, which is triolate, in southern Eyre Peninsula and in some of our South Australian, southern Victorian surveys. So this is a bit of a problem because we have to, the reliance on pre-emergent herbicides has increased of late, and with plants now developing Group J resistance, we've pretty much left with the Group K alternatives. And Sakura, while we've shown has been effective in controlling ryegrass, is only registered for wheat and pulse crops, and that leaves us very little options to work with when we're dealing with ryegrass infestations of barley crops. So I think well under the time limit. Thank you. Should I turn that off?
Questions for Mahima? Yes.
Hi.
What's known about capeweed resistance, capeweed sensitivity to 2,4-D, and then secondly, what rates were required in testing on capeweed?
So the question was what's known about the sensitivity or susceptibility of capeweed to 2,4-D and the rates that were used in our tests. I would have to handball that question off to Chris Preston, just because he did these experiments and it's still in the very preliminary stages, so we're about to repeat them again. But they weren't too high. I think the maximum was half a litre of 2,4-D.
More questions?
Yep.
Yes, sorry.
I was just wondering whether you knew the herbicide application history of the paraquat-resistant weeds, whether they've been used. Did paraquat to death, or whether it was...
Yeah, paraquat is used quite extensively in sugarcane crops, and this farmer had obviously been using it in a lot of his fields. And given that they've all come from the same region, we would assume that, it has been used quite extensively.
Any more questions for Mahima? Okay, thank you very much.
Thank you.
Now I'd like to invite the 5 speakers in this session to sit out the front. We'll have I've got a few general points that I think are relevant to further discussion, and I'm sure that you all have some as well. And while they're sitting down, I'll just get the notes. And Gary, is this part also recorded? In which case, how do we handle microphones? Okay. So we'll hand that back. Look, I've got a few things that came out of the talks for me were as follows. This session is on recent developments in the field. What's happening out there? For the fungi people, the first two, Fran and Andrew, we heard about the grains industry. We didn't hear anything about horticulture, and we just heard, so I guess that's something that would be of general interest, what's happening, and I suggest Fran would be someone who could take that up. Don't do it yet, I'll just give you some overview of comments and then we can discuss them further. Similarly for the insects, what sort of other, are there any other burning issues that you didn't, insects that you didn't handle that are out there that we should be thinking about just in general of what's going on in the field? With the herbicide, I think that was really covered very well, but perhaps there are other things that you'd like to say. So what else is happening? That's the first thing I thought about. The other thing are the assays and the sampling. I mean, we have sampling. We often take single fungal isolates, take them back to the lab and do things. So we want to have, how do you go single high throughput? Then, of course, the assays that you have. So we have the field surveys out there, what's happening, what a farmer can see in the crop. The in vitro assays that you bring back to the lab. And then, how well do they correlate? Angela Vanderwell is going to talk tomorrow. She works on blackleg and canola where she's using a high throughput assay where we're looking at fungicide resistance in the plant itself. And then as Steve Powell pointed out, well, we've got these target genes that we sequence, but maybe there are other things happening there, and we saw a great example that Grant brought out where, that wasn't quite what might have been expected. And, we have the ability, and this will come through, I guess, in the next slot, to talk about genetics and how you can track this. So, I don't know, maybe it's best if you start off, I don't want to dominate, which I clearly am at the moment. Are there other major sort of topics too that we could... Yes, Gary, please.
Well, I'm interested in each of the speakers reflected somewhat on our ability to practice change and address the resistance evolution issue. I'm just interested to sort of contrast. Grant made the point that they've really been quite successful in reducing the incidence. So and I think Andrew's point was we need to get the message out there about
Yes.
integrated management. How successful have you been?
Yes. No, that's a great point. And I guess also that'll come out a bit too in the last session today, the responsiveness of growers to this sort of advice and, how we actually give it. So perhaps can I start by asking Fran to give us a minute or so on perhaps obviously there's things like the wine industry, other industries, other issues that there are out there that may not have been covered.
Right. Thank you, Barbara. I had a slide in my presentation about grapes. Probably just went very briefly on that slide. I didn't want to steal the show from a presentation that will be happening tomorrow during the fungicide workshop. But basically, the situation in the grapes industry with regards to fungicide resistance, not in the field, because this is always in vitro, phenotypically and genotypically, is quite scary for botrytis. So we've got resistance to many modes of actions, as you have seen in this slide, if you remember, up to 6. And I'm pretty sure that there are more. So this is what we have tested. So in other countries, like for example in strawberries and botrytis happening in strawberries in the US, we've got up to 7, 8 modes of actions happening at the same time. So another advantage that we've got is that we can use a sort of resistance against the disease, because obviously the resistance normally happens at a cost, and that's a fitness cost. So we can use this to try to tackle disease or fight it back. So there are some strategies from the management point of view that we can deploy, right? But it's quite difficult, to adopt those strategies. We've got all the pathogens in the grape industry, obviously. For example, powdery mildew. So powdery mildew in the grape industry here in Australia is quite concerning, because it has developed resistance towards 2 different modes of actions at the same time, and this is something that we haven't seen anywhere in the world. All right. For DMI, so the fungicide targeting CYP51, resistance is just minimal. It's just we've got this precursor mutation that will prepare the structure of the target to further mutate. But in the case of strobilurins, we've got the famous G143A mutation quite basically everywhere.
Okay, so we've got grapes, we've got strawberries, horticultural crops. Are there any other comments from the floor about that, or if people see that there's some burning crop we've forgotten about? There isn't? Excellent. What about insecticide resistance out there? You covered the cotton, and Paul gave 2 examples, the green peach aphid and red-legged earth mite. Are there any sort of other burning issues in agriculture
well, I think,
or horticulture that are out there?
Western flower thrips, like the major pests go across lots of commodities, and we were talking about green peach aphid, which goes across many commodities, and certainly the aphids I was talking about go across many commodities, as does 2-spotted mite. Well, Western flower thrips is resistant to just about everything that's used against it. It will wipe out crops because it transmits viruses, so you get 100% crop loss. And because these viruses are sometimes hidden, you plant something else and then that gets wiped out, and then you plant something else and then that gets wiped out. So certainly in things like horticulture, they cause amazing damage. And because of their cryptic nature, they hide. They're very difficult to control. And it was, like I was working on it 10 years ago, and now just a little bit on the side, and it seems to be forgotten. but it's got potential to wreak enormous damage.
Okay.
Sure. I mean, I guess you've opened up a little bit of a can of worms.
Well, it's
starting
not
so
a
broad,
long
but
can
I mean,
of worms, a short one.
I mean, certainly. I mean, if we were talking about individual species, I mean, some of the other big ones that I guess are sort of no-brainers are things like diamondback moth and Helicoverpa midra.
Yes.
Again, similar to I guess what Grant's saying, across a whole variety of different commodities, and certainly both of those species with a very high propensity to develop resistance very quickly to chemicals when they come into the market. But I guess, of course, you know, we can talk about the species that we currently know have resistance, but of course, you know, the challenge for us also is to be forward-thinking and trying to predict as best as we can what might be on the horizon in terms of new resistances developing in species. And of course, there's you know, a lot of effort going into trying to develop frameworks to do such a thing. And I guess that's a challenge and a discussion for another day. But I guess, I mean, the point that I guess I didn't get to that perhaps I can make now is, and again, you know, Grant alluded to the role of neonicotinoids in cotton aphid. Certainly, from a grains perspective, you know, neonicotinoids are now widely used as seed treatments, obviously going across most canola, becoming more prevalent as a seed treatment on cereals and pulses as well. Obviously, you know, from a red-legged earth mite and green peach aphid perspective, if we're talking about the species that I covered this morning, already similar to cotton aphid, we're now starting to see the first evidence of neonicotinoid resistance in green peach aphids, which is obviously concerning, and certainly is not surprising given that we know this species has resistance to that chemical class overseas. And I guess from a selection pressure point of view, given that we, as I just mentioned, have neonicotinoids going out across very large landscapes as seed dressings in grains crops, of course, thinking about species like red-legged earth mites, there's a hell of a lot of selection pressure now being exerted on that species, as there are for others, to potentially, you know, evolve resistance to that chemical class. So, you know, I guess that's one that I raise because for us as a grains industry, we are heavily reliant on neonicotinoids as seed dressings for a number of different crop establishment pests. And as I mentioned, with something like red-legged earth mites, at the moment we have resistance to our 2 foliar sprays that are available to us in terms of the 2 chemical classes. The other chemical class that we rely upon is the neonicotinoids as a seed dressing. So, you know, there's certainly a need to start to monitor and to generate some baseline sensitivity data for that chemical group. Not to mention the fact that, of course, you know, what's happening overseas with that chemical group and the real possibility of that being withdrawn from markets anyway. So, certainly forward-thinking, I would suspect that there has to be a focus, and there certainly is within our group now starting to talk about that chemical class.
Thank you. Can I open it to the floor, perhaps, to make some general comments or queries? I mean, you know, I made the one about the sampling, the assays, etc. I think it's really broad. We could probably spend an hour talking on it, and I don't really want to start. It would never stop. But, you know, please, you're here to make comments and ask things of the speakers. Please do. Oh, yes. And, oh, well, that's fair enough. It's hard to, though.
I guess in terms of the integrated pest management and control strategies, if I've got this right, Paul, with the green peach aphid situation, one of the issues was that farmers used the wrong chemicals, which then got rid of beneficials, which allowed the green peach aphid to take over, and then we had this epidemic. So I guess one of the issues we face is that we have this great amount of information that we know from the lab side of things, knowing what resistance and what, you know, combinations work. How do we make sure farmers are getting that information and putting it into practice to sort of prevent that situation that happened with the green peach aphid last year?
So I guess I would certainly hope that, you know, I guess a lot of what you're talking about there is going to be picked up in the final session that I believe Steve is going to be talking in, because it's a critical component of everything that we're talking about today, is how do we get resistance management strategies firstly developed. I guess that's perhaps the easier part of the equation, but then how do we get, as you say, growers and advisors adopting strategies that we believe scientifically are sound, but of course what we hope to do develop those strategies in a very practical way in consultation with industry, so that the recommendations have every opportunity of being taken up. But of course, the question around taking up resistance management strategies is a social question as much as anything, and certainly is probably no different to growers taking up broader IWM, IPM type strategies, and to me they're one and the same anyway. So I don't have an answer as to how we achieve that. Clearly, I'm sure we'll get some fruitful discussion this afternoon, hopefully. But it's certainly something us as a group, the NERM group that I mentioned, we've grappled with. We believe we're developing some really valuable documents and really good recommendations, but are we seeing practice change? And that's a challenge. And of course, the next challenge is how do you then measure the success of your strategies if they are taken up? Again, that's I guess another level again. So,
Thank you.
Not quite sure I answered your question. I think I just diverted.
Other comments? Yes, please.
Yeah, all the isolates that we've tested so far, which is 200 odd isolates now across a couple of years, that historical set, and up to isolates that we've tested this year, none of them have shown the G143 mutation to convey resistance yet in the populations that we've tested so far. So yeah, so at the moment, the strobes are still effective in Australia.
That's a function of not being used, do you think?
Well, perhaps not being used in isolation as heavily as they have been in other countries where they've broken down. I mean, I think it's probably only a matter of time, really, particularly if there was anybody out there using them in isolation. And that's been the characteristic in the UK, and I believe in New Zealand as well, where there were products going out where the strobes were used in isolation, whereas we haven't had that situation here. And neither have we had the sort of pressure or the preferential use of strobes in those products that growers are using to target septoria. They've been primarily using products that haven't had a strobe in them, is my understanding. Nick might have a comment on that. He'd have a good feeling for the products that have primarily been used to target septoria over the last few years have not been predominantly containing a strobe.
I think that the point you make about strobes being used in isolation was certainly a factor in the UK instances of resistance, probably less so in New Zealand, where I think that the lesson was almost learned from the UK is going to be mixed.
Okay, so it did break down in the presence of the azoles. Yeah, okay, all right. It's been identified. I don't know that it's been widely distributed yet in the population, is my understanding.
It's been identified, but the mutants, as I understand it, that have been identified are carrying high fitness penalties in septoria.
Can I follow on from that about the sampling? You've looked at, say, 200 isolates that you've got from the field or a historical collection, etc. We need to see, obviously, correlations between what's happening in the lab, what's happening in the field. So how do we do a more high throughput way, and what numbers do we need to, for instance, have those historical isolates that you've got? There are obviously a few of them, but how high is the incidence of resistance or tolerance? You showed us in that lovely last slide an escape or whatever. But there's a dilemma there, isn't there, with this, how much you have to do and what the in vitro stuff really means.
Yeah, so I guess, with all of us, the sampling that we'd be doing for whether it's pathogens or insects or something, we're only taking a thimbleful out of the population. Personally, in the approach that I've been taking, we've been collecting structured population samples as frequently as we can, as well as random like transects through paddocks to start to unpack some of those population genetic issues for septoria. Is it enough? I mean, we're only at the beginning, so the high throughput nature of it, none of the techniques we're using I don't think are terribly high throughput other than the molecular ones, particularly phenotyping in the glasshouse or something like that, which we've been starting to do. Yeah, we're really limited by the number that we can push through there.
And I wasn't picking on you in particular, but it was just following on, because as you say, it's relevant to all the situations we've had today.
Yeah, I think it's particularly challenging for pathogens or insects that are out crossing. I mean, as septoria is. Yeah.
Just one last thing. We haven't heard in this general discussion anything much about herbicide resistance. You did a fabulous job covering it, but are there any burning questions about that? Because then we've got morning tea to follow. Nothing's burning? Oh, there is two burning people. Yes.
Just really quickly, I realise Chris isn't here, but maybe Paul, Graham and yourself might like to make a comment about marrying the insect resistance with the weed resistance and the excess of weeds in a landscape which leads to a build-up of pests.
Well, I think that was, weeds has been one of the main things that we've been trying to control. And a lot of the work was actually going and looking at pests around, weed species around cotton. And one of the worst weeds around cotton is cotton, because the transgenic cotton kind of escapes, and once it escapes, it's very hard to kill. And if you go looking at them for resistant individuals, they're very, very easy to find, and in some seasons, I was, most of the weed cotton was carrying resistant individuals, so that it's absolutely imperative that if you're going to control resistance, that you control harbourages around the crop that you're trying to manage, and if you don't do that, you're not going to be successful.
Okay, thank you very much to everybody, particularly our 5 speakers, and it's a great start to the meeting. So thanks to all the speakers.